Okay, everyone, welcome to the first part of our three-part series on connective tissue disorders. Today, we are going to be covering part one, connective tissue and hypermobility. But first, I want to do introductions. So thank you, everyone, again, for joining us today, either live or through this recorded presentation. For those of you who may not know me, my name is Rachel Baer. I'm the co-founder, director of education, and COO of Clover Genetics. I have a bachelor's of science in biological sciences and a minor in depth studies from the University of Cincinnati, and a master's of science in microbiology and cell science with a concentration on medical biochemistry and biotechnology from the University of Florida. So for my master's literature review, I compiled data from over 50 recent scientific publications and wrote an updated review on the state of the Ehlers-Danlos Syndromes and overlapping heritable connective tissue disorders, outlining the gaps in the molecular research and management recommendations. And I personally have both a diagnosis of a heritable connective tissue disorder and an autoimmune connective tissue disorder. So I'm particularly passionate about raising awareness for these different conditions and sharing in depth with the community about the impacts of them on the public health benefits to furthering education and research for those both diagnosed and undiagnosed patients living with connective tissue disorders. I am joined today by Clover Genetics genomics correspondent intern Ilham Abbasi, who is a current graduate student at the University of Toronto in her second year, completing her Masters of Human Genetics. So I'll pass over the mic to her to further introduce herself.
Hi, everyone. As Rachel said, I'm a master's student right now. I'm studying medical genomics. So learning a lot about the different disorders and how genomics all comes together in the pathology of these different disorders. And I'm a genomics correspondent here, so I've done some work putting together epilepsy resources for our website, and I'm here to help Rachel today with her presentation on connective tissue disorders.
Thank you, Ilham. So next, before we dive in, I want to emphasize our disclaimer here. We are not doctors or nurses. This information is all for informational purposes only. This is intended to provide broad knowledge of health based on current scientific research, not intended to be a substitute for professional medical advice, diagnosis, or treatment. You should always seek the advice of a physician before making any modifications to your healthcare regimen, diet, exercise, or treatment plan. And after reviewing this information, you can highlight things that stuck out with you and review them with your physician. But again, never delay seeking medical care for any concerns. And this is not a substitution for any medical recommendations. So today I'm going to give an overview of why what we're discussing today is pertinent to the overall picture before I begin to kind of break down the molecular components of connective tissue that we need to understand before truly understanding how these connective tissue disorders manifest. And then Ilham's going to outline different types of connective tissue disorders and how genetics plays a factor. And then I'll finish up by elaborating further on kind of the phenotypic presentation and quantification of hypermobility before we conclude and open the floor for discussion. So please keep in mind that the second part of this presentation is going to be taking place on July 31st at 1 p.m. Eastern Time, and we'll focus on the genetics and genetic testing utility for each of these conditions. An invitation to this event will be sent out after today's presentation, and the third part will take place in August on a date to be announced, where I'll provide my own personal experience with diagnosis. We'll continue to touch on the subject, but before we proceed, I just want to make sure we differentiate these characterizations. So connective tissue disorders, it's an umbrella term, where heritable connective tissue disorders, or HCTDs, live underneath that umbrella, but are also separated by specific genetic or heritable or inheritance patterns. And before we discuss the intricacies of the biological aspects, I think it's important to understand who the underserved patients are and why it's important to get a better understanding of this group of conditions. So connective tissue disorders can be difficult to diagnose, particularly for those with heritable or genetic components. When those red flags are missed by non-genetics providers, they seldom pursue genetic testing, which inevitably can leave patients without the appropriate medical intervention for sometimes decades before diagnosis. So the average number of physicians or providers that a heritable connective tissue disorder patient sees before receiving a referral to a genetic specialist is 3 to 4. So not only that, but specialists in the genetics realm willing to see patients with genetic disorder, connective tissue disorders are far and few between. Sometimes they don't accept new connective tissue patients, or they'll add them to a wait list for years, or they may require them to travel long distances, often to be met with little compassion, understanding, or answers. So that can vary for a lot of different reasons. That's not to say that these providers aren't wanting to help patients, but there are a lot of people that don't understand how to address these concerns, and so it can lead to these backlogs of the one provider that might be in that region that seems to be able to help, or it can make providers feel like they are not qualified, and so for the patient's own safety, they don't want to take them on as a new patient. Those who are unfamiliar with genetic conditions or lack access to reliable medical care might be plagued by progressive deterioration of their physical health over time. They may experiment with self-medicating supplements, either guided or experimental, pseudoscientific practices, or simply just living with debilitating symptoms and pain every day. This can be an attempt to cure a condition that has not been diagnosed, and it can continually exacerbate the mental health and chronic pain symptoms of those conditions as they tend to get worse. So in summary, it's important because those living with heritable connective tissue disorders are plagued with constant use of the healthcare system, and they're often met with a great deal of provider inexperience, lack of understanding of the multitude of symptoms that must be managed with highly specific recommendations. And so the goal is to raise awareness. From a genetic counseling perspective, genetic counseling, most heritable connective tissue disorders can only be diagnosed through next generation sequencing. Ultimately, this can aid in improving the understanding of certain genetic conditions, identifying new variants that cause the disease, or opportunities to make specific drug therapies that target the root cause of genetic condition. Another reason genetic counseling can be useful for these patients is because pharmacogenomics testing can also help to curb negative side effects, even for undiagnosed patients, by helping them understand which drugs that are often going to be utilized should or should not be approached. So that could be for pain, blood pressure, anesthesia, symptoms of depression, other common symptoms. And another big factor is that there are specific genetic heritable connective tissue disorders associated with high risks for fatal arterial or organ ruptures. And those can be prevented with specific drugs or prepared for in advance through lifestyle changes that reduce the risk for those chances. Okay, so what is connective tissue? There are many different ways that connective tissue can be classified. It can be based on the actual organization of the cells, as we see in the image here, or it can be based on what the function of the connective tissue is, or even based on the tissue type, which can make it a really hard topic to Google, especially as a patient, if there's somebody trying to understand what connective tissue is. They can be led down multiple routes that can all seem correct, but without the understanding of the cell types, they can blend together. So here we're reviewing the organization of cells and the relationship between the structures to one another. When most people refer to connective tissue, they're referring to loose and dense connective tissue. So these here. These are usually grouped together as connective tissue proper. And then connective tissue proper can be further divided into loose or areolar tissue and dense tissue. So dense tissue, and then again, it gets more complicated. Dense connective tissue proper can be regular or irregular. Regular dense tissue is made up of parallel fibers, like tendons and ligaments that provide unidirectional strength. Whereas irregular dense tissue contains multidirectional fibers. So as you can see in the picture, for multidirectional strength like skin, loose connective tissue functions to hold together organs, anatomic structures, and other tissues in place. And so it tends to have really big spaces between the fibers surrounded by a gelatinous substance that allows for fluid movement. So in this visualization, there's another way to classify it based on the role that the connective tissue plays in the body rather than the actual cells. So outside of connective tissue proper, the specialized forms can include the fluid, fascia, elastic, reticular fibers, bones, cartilage, fat, membranes, all with highly specified purposes. And the functions of the connective tissue can be summarized into 7 general categories. So the first one is enclosing and separating. So like hollow organs of the stomach, intestines, uterus, bladder, valves of the heart, which again could go back to those arterial ruptures that we mentioned if there's any genetic change that alters the structure of the valves of the heart. The next one is connecting. So connecting different types of tissue to one to another, like ligaments attaching bone to bone. Supporting in movement. Bones are an obvious example of a critical aspect of connective tissue, along with the muscle required for the movement of bones. Storage, so energy reserves like triglycerides, minerals, biomolecules can all be stored in adipose tissue and other locations. Cushioning and insulation. Oh, sorry, it was hidden on my screen. That it's similar. Fat provides insulation from cold and cushions areas of impact. So like the balls of your feet or your butt, where your bony pelvic bones would otherwise hurt every time when you sat down. But keep in mind, so we just talked about fat or adipose tissue in 2 different contexts. So multiple roles, but describing one cell type. So another way that it can become confusing when you're talking about the classifications. Transporting. So this can include fluids like blood and lymph. Generally, blood is considered a type of connective tissue as it carries oxygen, nutrients, and biochemicals like antibodies and important things to transport away like waste. However, like a blood disorder is not specifically a connective tissue disorder. And some individuals or places of classification might completely exclude blood when they're talking about types of connective tissue, just to make sure that it's differentiated, because there are also other cells and other things within blood that don't stem from the same genetic source. So, lastly, protecting. So protecting delicate organs either with cushioning, fluid, support, skin, membranes, or other means. So connective tissue itself, it's comprised of different cells, different fibers, and ground substance. So when an embryo begins to develop, there are certain types of cells or cell lines that start with just a few options. And then as the embryo grows, or it branches out to different cells that can develop. So the types of cells we're discussing specifically are called mesenchymal cells. So these are the embryological cells that differentiate into connective tissue. So that's kind of what I meant. Within blood, there are other cells that aren't necessarily from mesenchymal cells, which is another reason it can be separated a little bit. As adults, certain conditions that involve the cell line origin may be more specifically impacted than other cell types. So while all cell types make up our bodies, there are specific types of cells more associated and more involved in the function of connective tissue. So fibers, like collagen fibers, form triple helical structures between one another. It provides durability. Irregular collagen or elastic fibers that cause some of these connective tissue disorders we'll discuss play a really important role in the structural integrity of the body by maintaining this effective binding with the strong fibers. And as you can see, the lines of the fibers are widely dispersed across this image. So they do create kind of a backbone. And then the space between the cells and fibers is filled with ground substance, or which is comprised, or which is part of the extracellular matrix, or the ECM. As noted, thousands of molecules all at play surrounding the connective tissue cells and fibers. And the ECM also creates a barrier for the invasion of pathogens, provides an exchange space for other molecules, fluids, nutrients. There can be immune molecules, growth factors, and other compounds that help ensure homeostasis within the tissue, including the breakdown of cellular waste. So for either fully or partially functional collagen molecules that get released into this extracellular matrix, there are additional modifications that further stabilize that triple helix. So if that doesn't happen, there can be consequences. Ground substance is a very general definition. We won't go too much in depth, honestly. It's important just to point out that originally connective tissue disorders, or the genes specific for collagen producing fibers, were thought to be the only genes that really mattered or were relevant. But it's become clear that these molecules within the extracellular matrix have to work. Even the most durable collagen fibers released into a dysfunctional extracellular matrix are going to fail to mature into healthy connective tissue. So, what is connective tissue? We've kind of figured that out at this point, at least a little bit. The mesenchymal cells are the specific cells that differentiate into the various components, but the specific cells that they differentiate into are the fibroblasts, the adipose cells, plasma cells, lymphocytes, leukocytes, mast cells, macrophages. So fibroblasts, to start, they create and maintain a matrix of connective tissue fibers. There are also specialized myofibroblasts that kind of repair tissue and can create scar tissue, which will be a big factor in a lot of connective tissue disorders, is irregular scar tissue. Adipose cells, we've already discussed, are fat cells. The way that they can play a role in pathogenicity is variable. Plasma cells and leukocytes, plasma cells, lymphocytes, and leukocytes are often involved in cellular transport, especially immune cells that are responding to inflammation or other chemical signals. There's a lot of inflammation generally involved in these conditions. Mast cells reside within the tissue and they're cell specific. They induce allergic responses. So keep that in mind when you're reflecting on the association of connective tissue disorders and increased allergic responses in mast cell activation syndrome and/or disorders. And macrophages. So macrophages, they're in each tissue type with specialized roles in the immune system. They recognize and digest harmful pathogens and cellular waste. And this can be really important in these conditions because cellular waste is naturally occurring, but with some of these genetic conditions, they can be manufacturing an abnormal protein that can't be used. And it can be mass amounts of this cellular waste that builds up in a way that the body has trouble managing without producing those symptoms. Okay. That being said, I will go ahead and turn things over to Ilham.
Thanks, Rachel. So now that we've explored the cells and basic anatomy and physiology of these different microscopic components, it's really important that we emphasize the role of fibers and differentiate between the three core categories. So there's collagen, elastic, and reticular fibers. And the latter we're going to cover on the next slide. So to start off, collagen fibers are made up of the protein collagen, which is characterized by a triple helix structure. So these can split and join back together in different areas to form a 3D meshwork. So in the figure here, you can see that these are those thicker pink lines that you see. And there's an estimated 28 different types of collagen in humans and 43 different genes that play a role in collagen formation. And this really allows collagen to be extremely versatile because different strands of collagen that form a triple helix together will be designated for specific functions. And collagen 1 specifically is the most abundant structural protein across vertebrate species. And so the natural structure of collagen emphasizes the importance of conserving its rigidity to perform its critical structural role in bone, dermis, cornea, and tendons. And then unlike collagen, which is really there for maintaining rigidity and strength, we have elastic fibers that are there for flexibility and the ability to bounce back to its original shape. So in the figure, you can see that these are those thinner sort of black lines that you see. And you can see that they're very structurally different to collagen. And elastic fibers are made up of elastin, which provides elasticity, and fibrillin, which provides structural support. And together they form this fiber network that can be stretched and recoiled without damage. And then lastly, in the figure, you see these little round spots. These are the fibroblast nuclei, which contain the instructions for creating these fibers. So reticular fibers are secreted by fibroblasts called reticular cells, and these fibers are composed of thin type 3 collagen strands that come together to form a supporting mesh network called reticulin. So reticulin supports soft tissue found throughout the body, and this includes the liver and bone, and it also forms structural frameworks of most organs in the lymphatic system. And although they are composed of collagen, they're not to be confused with collagen fibers. So reticular fibers aren't as bundled and ordered as collagen fibers, and you can easily differentiate between the two based on their different reactions to chemical stains. And reticular fibers are also a lot more narrow, and they don't form thick filaments, and they also have a higher carbohydrate content. So just to put this into context a little bit, a connective tissue disorder called vascular Ehlers-Danlos Syndrome, or VEDS, which is also formerly known as type 4 EDS, is an example of a connective tissue disorder that arises from autosomal dominant mutations in the COL3A1 gene. And this gene encodes the pro-alpha 1 chains of type 3 collagen strands. And so because of that, this can lead to difficulties in assembling reticular fibers. And the synthesis pathway of reticular fibers is quite complex, and we're not going to be getting into all of it today. But what I want to highlight here is that there are 3 different parts of the reticular fiber synthesis pathway that can be disrupted by these mutations. So first is the production of the pro-alpha 1 chains. Second is the release of a precursor molecule called procollagen, which is made up of pro-alpha 1 chains. And lastly is a synthesis of collagen type 3 molecules, which are made using procollagen. So you can see there are different parts of the pathway that get disrupted, and it's sort of this chain reaction where the entire synthesis of reticular fibers is now impaired. So now we're going to move into talking about what heritable connective tissue disorders are, or HCTDs, and I'm going to give you guys some more background on them. So when it comes to connective tissue disorders, there are so many aspects of genetics that come into play that can lead to such a broad presentation of phenotypes and symptoms. And a lot of the guidance and recommendations made for patients are based on confirmatory genetic testing positive family histories, or the simple best practices described as a blanket standard of care. Now, this is beneficial in many ways, and we should never make recommendations based on theoretical knowledge or guesswork. But with that said, the more patients, providers, and allied health care professionals understand about the biological and full body impact of these conditions, the better we can grasp the impact of this condition on patients and further focus research to address the needs of this underserved patient population. So just keep this in mind as we explore more about the molecular biology and biochemistry of the tissue and how patients might experience symptoms of these processes and what impacts these have on phenotypes. And so this is important because there's a lack of understanding across the spectrum of providers that these patients are seeing. So oftentimes, non-genetics providers can miss connective tissue disorder red flags, which means that these patients aren't receiving any clinical genetic testing or diagnosis. And because of this, HCTDs are likely underdiagnosed, and the prevalence of these disorders is probably higher than what's been reported. So what connective tissue disorders come down to are a lack of stable tissue support in any of the components we've described so far. So reduced tissue integrity leads to impaired function that can cause diverse pathogenic phenotypes, which can vary based on the nature and the individual's diagnosis and genetic makeup. So there are over 200 connective tissue disorders, and they can range from being very commonly occurring, such as hypermobility, to being more rare inherited disorders that have more severe phenotypes. So some connective tissue disorders are caused by changes in genes that affect the production of connective tissue. So these are HCTDs, and they're typically rare. Next, there are people who are genetically susceptible to having something in the environment trigger a connective tissue disorder. So, for example, these can be autoimmune disorders that have both genetic and environmental components to them. Next, you can have acquired connective tissue disorders. So the main example of this is scurvy, which is caused by a lack of vitamin C, and it leads to an inability to make collagen. And then finally, some connective tissue disorders still have unknown causes. And so although there are so many different types and causes of connective tissue disorders, we're going to be focusing in on the heritable ones from now on. So because of the diversity of heritable connective tissue disorders, it can become very difficult to diagnose them. And what complicates these disorders is that about 36 of them have overlapping features like hypermobility. So not only that, but some of these disorders fall under the umbrella of a broader disorder, such as EDS, which is a group of about 14 different disorders. And so some HCTDs have highly specific phenotypic presentations that can allow for them to be diagnosed without the use of molecular testing. But with other HCTDs, such as the EDSs, there's poor genotype to phenotype correlation, and there's so much overlap with other HCTDs that molecular evidence is the best way to differentiate which range of phenotypes might be impacting them. And so currently there's over 67 confirmed genes that have known causative pathogenic variants that present with variable severity and phenotypic expressions. But why is this? So there's two main reasons for this. The first one is pleiotropism, and the second one is variable expressivity. So pleiotropism is a universal feature of HCTDs because it describes how variation in a single gene can manifest itself in multiple organ systems and in many different ways. So the specific organs affected and symptoms occurring are based on a number of factors, including the gene's expression during embryological development and throughout childhood. And so we can see how this actually impacts patients with HCTDs. Pleiotropy is a fundamental genetics concept that plays a role in the expression of genes and can also be understood through the impact of one gene influencing multiple phenotypes and therefore multiple body systems. So a good example of this is Marfan Syndrome. It's a heritable connective tissue disorder that's typically caused by autosomal dominant variants in the FBN1 gene. And this can influence multiple different traits, including the cardiovascular system, the skeletal system, and vision. And this FBN1 gene encodes for fibrillin 1 protein, which forms elastic fibers. And so, as we talked about earlier, elastic fibers are expressed in multiple different tissues. And so, because of that, this can cause multiple body systems to be impacted by this one genetic change. And the second fundamental concept we need to understand is variable expressivity. So variable expressivity is a term that's used to describe the different degrees of severity of symptoms experienced by individuals that might be carrying the same pathogenic variant. So in other words, some people have worse symptoms than others, even though they have the same genetic change. So for example, two family members could have the exact same pathogenic genetic change in the same gene, but one family member might experience very mild symptoms while the other family member experiences very severe symptoms of the same disorder. And so in addition to pleiotropy, variable expressivity is very common in Marfan syndrome. So while some individuals with Marfan syndrome have fewer life-threatening symptoms like having a tall and thin build, abnormally long arms, legs, and fingers, other individuals can have more life-threatening symptoms, including cardiovascular disease and aortic rupturing. And so because of this, diagnosing and treating people with HCTDs can be a long and difficult journey that requires genetic testing for truly informed medical management. And more often than not, it's those with more severe symptoms that are being offered this genetic testing, meaning there's a significant underrepresentation of the amount of people who have heritable connective tissue disorders, but they're just presenting with milder symptoms, and so they're not being tested. And I'm going to return the presentation now to Rachel to finish off with an explanation on hypermobility and how it connects to this group of disorders.
Thank you so much, Ilham, for those explanations and that background surrounding heritable connective tissue disorders. So for this last section, I want to elaborate on hypermobility and kind of what it means in a clinical setting. So hypermobility is a measurable trait. That being said, it isn't always measured correctly. So this chart is available for download from the Ehlers-Danlos Society's website as a tool to help understand the standard scoring system for hypermobility. Currently, the Beighton scoring system is the Beighton scale, sometimes called, is the current system that measures a specific series of joints on a 9-point scale to determine if a person has a higher than average hypermobility in their joints. Without additional symptoms like pain, dislocations, soft tissue tearing, or ruptures, hypermobility on its own is not a pathogenic trait. So, when you hear about contortionists, not everybody that's a contortionist has a hypermobility syndrome. But when the person has higher than normal hypermobility, and then they also are starting to have symptoms that, or, tearing ligaments, things like that, that is when it needs further investigation. So, and significant hypermobility generally can be something that indicates further investigation as well. So this scale is a little bit controversial currently as it excludes several joints, and it really evaluates the mobility of the knuckle of both pinkies at the base of each thumb, the elbows, the knees, and the spine. So it doesn't include any others. So like the hips or neck, other things like that. So if you look at the photo, you can see how standard and quick that this examination can appear to be. You have this done at your primary care doctor's office. You could do it yourself at home. But proper geneticists and physicians that understand the value of this test might be and should be very thorough to use tools like calipers to measure the angles with very high specificity rather than leaving it up to their own judgment. So there are also other recommendations when, to take this test where there's a certain amount of time that the patient is supposed to relax in a certain position before they do this, not having them go into this like straight from heavy exercise. And so there are some other factors that are important. But the next important thing is to understand why hypermobility is a sign of these conditions and how to recognize the syndrome beyond just this bite and score. Because it's important to know that there have been additions made to this criteria that, because not everybody is comfortable with it, because there can be additions of points for what's called historical hypermobility, because there can be patients who were at one time able to do all of these hyperextensions, but maybe due to injury or age can no longer. And so that is an important factor that needs to be taken into account by the scoring physician. So again, we've talked about what hypermobility is based on the scale and the testing, but there are phases of hypermobility that can start to indicate the progression of a syndrome. So in youth, first of all, a lot of children are naturally very flexible. So some geneticists might not even be willing to see children, under the age of like 8, if they're not particular, like, if there are other symptoms and whatnot, because it would be considered benign hypermobility. And as children are growing and their joints are stabilizing, they tend to be a little more flexible. I can't do a cartwheel anymore. I used to be able to do one with ease, things like that. So during youth, patients that have hypermobility might show them off as party tricks, like how we think of like contortionists, or if you ever had a friend that could, twist their arms around really funny, or things like that. So it's not that it hurts, it's just something that they start to notice that they can do in their children. So as children are developing, it can kind of maybe prevent a physician from taking much note of those, because it kind of borders on a syndromic presentation. But typically around puberty, there are these kind of mysterious symptoms that begin to appear. So suddenly maybe there are joints that are dislocating or subluxating, which is like a partial dislocation, or you've torn a lot of ligaments or joints, or you're having problems with fatigue or migraines or a myriad of other symptoms. This is the aim of a lot of points for diagnoses, is like in this area of puberty. So a lot of clinics for connective tissue disorders are often for children. And so those who do not get diagnosed in their adolescence or young adulthood can move from this kind of mysterious symptom stage into the pain and injury stage, which can be early 20s through 40s. So daily wear and tear on joints and ligaments without that quick natural recovery that a typical non-hypermobile person would experience can start to tell the body or signal to your nervous system in a constantly active state saying, hey, there's pain, there's something wrong in this joint. And so with the surrounding muscles can begin to stiffen, and it can be kind of consistent with central sensitization or fibromyalgia. So in other words, chronic widespread pain. And the stigma around those diagnoses in particular is high due to the invisible aspect of the pain without obvious outward disease expression. So sometimes patients are diagnosed with pain disorders before they're ever diagnosed with a hypermobility disorder. And then again, if it is untreated past that earlier stage of the pain and injury, stiffness begins to occur in untreated or chronically injured patients. So as stages of damage and improper healing have accumulated, muscles around the body might tighten in a protective state, while the joints and ligaments might be weak and loose, and that can make patients prone to further damage. And so if patients are not diagnosed yet in these stages, which can be variable, this stiffness stage, that might happen earlier than early 20s, even if it's a very severe stage, like we talked about, or like Ilham talked about with the variable expressivity. But once patients reach the stiffness stage, they will often not score high enough on the Beighton scale or the Beighton score to flag a hypermobility disorder, even if they were formerly very hypermobile. And the scale, and like I mentioned, it allows for a historical hypermobility to add 1 point, but not to add all of the points back. So that can complicate things further for older patients that are trying to get diagnoses. And the last stage, which, this recovery and treatment plan, ASAP, so that hopefully maybe would start right at that mysterious symptom stage, or hopefully, before the pain and injury. But if not, the very last thing would be, and not very last thing, really this is the start, is you identify the appropriate testing to get the clearest extent of a patient's hypermobility symptoms. Any outlying medical issues, comorbidities sometimes have to be sorted out. Some conditions require, once they are diagnosed, might require preventative measures. Others can be managed with screenings, lifestyle changes, but management is often considered effective once a patient can solidify their care team, identify their best routine, and gains the required supportive care or community that allows them to follow their individual recommendations. So again, this whole thing is going to vary dramatically depending on the condition. And so when we talk about some examples of hypermobility syndromes, not all heritable connective tissue disorders have a hypermobility component necessarily, or they might not have a severe hypermobility component, or it might be isolated to a certain joint or to just the skin and not the joints. So we're going to explore each of these further in the next part of the series, but understanding this is kind of a key point so that you can also understand there are many different presentations of hypermobility syndromes and heritable connective tissue disorders. And so the complexity of this, keeping patients from getting support, can further that disparity that we talked about earlier. So one example I'll give before we kind of summarize the overarching effect on the body is that, as we've already kind of mentioned further back, we talked about the importance of how that triple helix of collagen 1 remains rigid. So on the left, first, this is a graphic I created using 3D models to compare kind of typical collagen 1 triple helix, which is on the left, to the altered collagen 1 triple helix on the right. So visually, you'll see that the tightness of the helix and the closeness of the molecules to one another on the left side, they're a lot closer, and it's negatively impacted on the right side, where this pattern of amino acids has been altered disrupted in the DNA sequence. So this is one of the mechanisms that is kind of common in several collagen disorders. That includes some forms of Ehlers-Danlos Syndrome, as well as some forms of muscular dystrophy. So again, I can go further into detail about these amino acids and whatnot when we have the next part. But the main takeaway here is just looking at those images and seeing that the slight alteration in the pattern completely dislodges this really tightly knit helices from these 3 strands from one another. So this comes back to just why it's important to recognize them. So teaching providers to recognize the multisystemic presentation and how to recommend them to the best specialists for the appropriate genetic testing or just regular testing is critical to getting them the correct diagnoses. Because again, reflecting back to the other types of connective tissue disorders, sometimes they can overlap with autoimmune disorders where it can be unclear which a person has. We've explored the concept of pleiotropy and how it can cause multiple different symptoms for patients in different systems, and that symptoms across the whole body can cause patients a great deal of anxiety, fear, pain, discomfort. And so when we look at how connected that these cells are within the body, it makes it clearer that a multisystemic presentation is a large clue pointing toward the need for a more thorough and holistic assessment. And then ultimately knowing the different ways that hypermobility impacts the body allows the provider to be more specific and personalized with recommendations for hypermobile patients that might be acutely ill or chronically ill with other conditions, undergoing treatment for cancer, using immunosuppressants during menstruation, planning for or recovering from pregnancy or menopause, those who are cognitively impaired or reaching elderly stages of life and might have increased risk for tissue fragility, all kinds of different things that need to be kind of customized to the patient based on their individual presentation of their condition. So this is an immense topic. So to summarize what we've covered today, this is just the groundwork for furthering your understanding of each of the heritable connective tissue disorders to be discussed next month. But to summarize, connective tissue patients face health disparities. We got to increase understanding for healthcare professionals. Connective tissue plays a role in every system in our body. One gene impacts multiple organ systems. Severity doesn't always align to the genetic variant patient to patient. Hypermobility can be benign or a symptom of something larger. Disorders causing tissue fragility can lead to more severe long-term consequences if you don't identify them earlier. And clinical genetic testing is often the only way to definitively diagnose patients. So, all that being said, here are some of the many sources that we have used, and that concludes part one of our three-part presentation. So I am now available for questions. Oh, yeah, Aishwarya, go ahead.
Hi, so is hypomobility a symptom in some connective tissue disorders?
That's an interesting question. So hypomobility is not really the terminology you would use. That would be more like different categorizations of stiffness, or sometimes contractures, where muscle rigidity can cause joints or certain things not to move. You know, the more I think, the more broadly I'm like thinking of different examples in my head. I just wouldn't describe it as hypomobility. I think there are different clinical terms that could be used, and then that would lead us to whichever answer. So kind of, yes, it would determine, it would be a symptom, but it might be a symptom of something that could overlap, or for example, like arthritis can cause joints to not move as well, or certain forms of muscular dystrophy can cause like stiffness, contractures, and things like that. Does that answer your question, Aishwarya?
Yes, thank you.
Yeah. And Juliana, your question in the chat. So as far as if there are specific criteria to qualify for genetic testing, it depends if you mean to be covered by insurance or if you mean by your physician. It can be really complicated, and it can depend on what kind of support you have. However, a regular primary care physician should be able to refer any patient to genetic counseling or to a geneticist to be evaluated if their patients are suspected to meet any of these. Like, for example, a primary care might do the bite and scale tests in their office, even though they're not that familiar with it. They know how to do it enough. And if they feel like it was high enough, then they might refer to geneticists. But as far as the criteria to qualify for genetic testing, there are so many different heritable connective tissue disorders that it would be it would depend. So when it is a baby, let's say, if a baby presents with symptoms or a child presents with symptoms, often A, insurance will cover that kind of testing, and B, the age of presentation is going to indicate whether or not testing is appropriate, and then that will also guide which conditions make the most sense. When it comes to things like adult onset connective tissue disorders or heritable connective tissue disorders, I think that it is much harder to get insurance to cover unless you meet a very specific criteria for some of the extremely like devastating consequence forms. So for example, like vascular Ehlers-Danlos Syndrome, where you can have ruptures of, you know, parts of your heart, or that same thing can happen with, you know, like Marfan Syndrome. If there is a strong family history and there's maybe even like an echocardiogram or some testing that, or some not genetic testing you've had that indicates or supports that you might have one of these genetic conditions, then insurance might cover it. Or if there's, you know, a family history in general, but it really does vary based on the age of the patient, the family history, and the presentation, I would say. However, they have connective tissue panels, like Invitae has one, that if you talk to a genetic counselor and the genetic counselor, like for example, we see patients all the time at Clover Genetics that maybe feel like, you know, I feel like I've had this my whole life. I have these symptoms, you know, I have a rheumatologist and, you know, they're not sure. Maybe the rheumatologist referred them. If it makes sense, then a connective tissue panel, often out of pocket from certain companies, can just be a few $100s, or they might have payment plans, or it might be covered by their insurance, depending. And so really it's if you have a doctor that signs off on it and agrees that it makes sense, and a genetic counselor, hopefully on your team, that's recommending the specific ones, then I would say that's all that would be required. But things vary. Yeah, sure. And when we talk about the next one in part 2, and I'll move from this boring sources page, we'll talk more specifically about the different types of EDSs. Like for example, one of the forms of EDS, classic EDS, there's a sign, but if you can touch your tongue to the tip of your nose, I think it's something like 5% of people that don't have classical EDS can do it, but 95% of people with classical EDS can. There are some things like that where if you can do those things, then testing might be, you know, like check that box. But because of this variable expressivity, when it comes to like qualifying criteria, like you can add that to your evidence, like as a provider or genetic counselor, and say, oh, my patient had this sign. Whoever writes the qualifications has to be really clear with, okay, what do we know as a fact versus like okay, this adds evidence, but it doesn't indicate the condition. So that can be a complicated subject for sure, but we'll explore further. Okay. I want to thank Ilham again very much for helping me put this together. I mentioned to her earlier, I've been working on putting this together since I wrote my really long paper 2 years ago, and realized that this needed to be broken down into sections in order to truly convey all the different components of it. So thank you, Ilham, for your help with this, and I'm looking forward to our next part, which